First, I really would like to thank Thomas and Bill and also Gonan to organize these fascinating events. And to be honest, this is the first time I gave a 90 minutes talk, right? So I would ask your like a cooperation to stop me as much as possible. Otherwise, I think we're going to take a coffee break after my talk. So my lab is interested in using single molecule imaging techniques, just as Thomas said, to study the form and the function of the 3D genome. And to just get the first most frequently asked question out of your mind is, where is Janelia Research Campus? So we are actually at Northern Virginia, a town called Ashburn. So it's right next to the Potomac River. So we are not far away from the Washington, D.C. If you drive 45 minutes, you can go to White House to protest sometimes, right? So the nearest institute is NIH, and the next nearest one is Johns Hopkins University. And so we have around 50. Oh, and also University of Maryland. And we do have a very nice single molecule community because of TJ. He organizes called the Chesapeake Bay single molecule meeting. So we usually have every year all the people, single molecule people along the Chesapeake Bay will get together and exchange what they find in the lab, which is quite exciting and a very close-knit community. The same as like Janelia, so we have around 50 labs, and every lab have very small number of post or trainees. So usually range from 2 to 6. Even you are a Nobel Prize winner, you can only, at the most, you can get 6 people. So they really have the policy to keep the labs small. So my lab is one of the 50 labs, and I think Bill and Thomas all visited like Janelia before. So we have a huge building hosting all the labs, and the building is a half-mile long. It's all made of glass, so you can a lot of sunlight. So my lab is one of the 50 labs, and I like philosophically, I always view my lab as a curious kid with a question in mind. And sometimes we don't have tools to address that question, so we build our own tool in hope that we can get that question addressed. But I think sometimes even better is based on the new observation we made, we actually find a bigger puzzle which demand us to build a better tool. My lab is also committed to share all the new imaging strategies and tools we build with the wider scientific community. So in today's talk, you probably will see several iterations of this going on in my lab. So my lab is interested in a fundamental question is how a single genome generates numerous cell types in our body through space and time. And one fertilized egg carrying a mix of mom and dad's DNA is able to divide and transform to around 26 trillion cells and forming hundreds of cell types in our body just under the period of 10 months. So my lab is aimed at developing imaging techniques allow us to visualize the molecular regulations underlying this process. So this is a particularly difficult problem from the imaging perspective because we are dealing with these nanoscale building blocks like proteins, RNA, and DNA. All these components, they organize to a micron range nuclear architecture and ultimately instruct the formation of tissues and organs in our bodies. And looking through the time dimension, we observe these molecular interactions occurring in the millisecond and even faster we couldn't capture, and all the way extended to lifelong physiological changes during the animal development and also in the adult tissues. So in today's talk, I will briefly talk about 3 groups of imaging techniques that we have been developing to gain access to each of these windows in space and time. So before I go into single particle tracking, I always want to talk about what's the earliest observation of single particle tracking. So there are some debate, but in my opinion, this a Scottish botanist, Robert Brown. So he observes these very interesting phenomena as a pollen. So he put the pollen from the flowers into the water, and the pollen dance around. At that time, he reported this finding, and people speculate maybe the pollen have life, so they can dance around, and then they boiled the pollen, and the pollen still dance around, and they use charcoal particles, which are definitely dead. They can still dance around. So people have going into debate with this phenomena like 100 years. And another thing people find is that the smaller the particle, actually they can diffuse much faster. So not until about 100 years later, and two brilliant physicists like Albert Einstein and James Perrin actually figured this out, and this phenomena is named as a Brownian motion. So basically, the water molecules combine bigger particles in the water and just push them around. And the smaller the particle is, the faster and the more stochastic this like diffusion is. And another, I think, quite important concept in microscopy is that we are talking about light microscopy with like a visible light. We're talking about the resolution. And we always have a limit about around 200 nanometer. So this is much smaller than the size of the pollen, but this diffraction limit is much bigger than a lot of these biological molecules, like lipids, protein, and even virus. And these particles actually below the like a diffraction limit. So the diffraction limit is being discovered by, like the protein molecules generally have a size of 2 to 5 nanometer. So the resolution limit, also called Abbe diffraction limit, is being described by Ernest Abbe in 1873. So at that time, he worked with Carl Zeiss. And so what he finds is that no matter how small the emitter is, if you look under the microscope, it always have a finite size. So what, and the diameter of this called area disk is roughly half of the imaging wavelengths. So what this means is that if you have two objects that is closer than the Abbe diffraction limit, and then we couldn't tell these two molecules apart anymore. And this is a quite important concept that also it's a limit that is really restrict what kind of detailed information we can get. And this diffraction limit has been hanging around for a really long time until two of my colleagues at Janelia, Eric Badzik and Harold Hess, and they are early pioneers. Basically, they worked in Bayer Labs in AT&T. So they have been working with single molecules their whole life, I think. And then one of the very simple mathematical concept is that although we have a diffraction limited spot, and we have this point spread function, if we fit a Gaussian to the point spread function, and we determine the centroid, and so we can actually mathematically, we have a much higher localization precision, about 5 to 20 nanometer. So back in, I think, 1995, right before Eric gave up science, I think at that time he gave up the science in 1995. He published a paper without any data. Basically, he proposed a very dramatic idea, right? So as we know, a lot of these biological molecules are packed within the diffraction limit. So what Eric proposed is somehow if we can separate those molecules in a third physical dimension. At that time, he proposed using spectrum properties, or in another word, color. You can separate the molecules and localize them one by one, and we can actually achieve much higher resolution than the diffraction limit image. I think because of this very obscure paper at that time, he actually won Nobel Prize in 2014, Nobel Prize for the chemistry. And so this idea, it didn't take a fly until 10 years later when people discovered this photoactivatable protein and the dyes. Basically, instead of separating molecules by colors, we can separate these molecules in time and reconstruct a super resolution image. And here, just give you a very simple simulation experiment. So you can see if we stochastically activate those molecules one by one, localize them in each frame, and we accumulate these localizations, we can get a much higher resolution image than the diffraction limit image. And this is one of the earliest palm, what they call is photoactivatable light microscopy palm image looking at these ribosome proteins in bacteria, I think at that time. Okay. So, so my lab, when I joined Janelia, I think my lab is really trying to extend this single molecule observation in live cells. And so we wanted to really watch how molecules move and how they do their job. And so this is very challenging, I think, until people find a tag. It's called halo tag. So this halo tag is a self-labeling tag. So if you fuse that to a protein, it can be covalently conjugated to a synthetic ligand with chlorophyll. Because the chlorophyll is so much brighter than the GFP, so which makes the single particle tracking cells feasible. So and one of the first question, yeah.
Protein, which is fused to the protein.
Yes.
What is the fluorophore? What is it?
So the fluorophore basically, this is, I think it's a dehydrogenase. This halo tag is dehydrogenase is in which species, I don't remember, but they can react with fatty acid, some linker. And it's basically the reaction is covalent. Once it reacts, it's covalently conjugate. And then what you can do is you can put actually different modalities to the other end of these fatty acid chains. And then you can put fluorophores, and some people also put, I think, those like a biotin ligand, they can pull down proteins. So it's actually a multifunctional tag that is quite useful. No, it's orthogonal. I think it's from a like bacteria. It's very specific to that species. Also very, I don't know exactly. Yeah, sure. I need to read through, I think, more carefully about the paper, how that, but it works really well in our hand. So we can, I think another advantage is that we can really tune the labeling density by just using a very trace amount of ligand. So we can get sparse labeling. Not like GFP, if you express a lot, a lot of copies will be within the diffraction limit. You couldn't tell a single molecule apart. The nice thing about halo tag, because it have two parts, the halo tag always fused to the protein, and you can decide what kind of concentration you use to label. So which you can really control what kind of sparsity you want, right? So to really tune to a single molecule region allow us to observe single molecules. So I think that's another advantage to have a two-part like a system. That's a real. There are some dyes like that. Yeah. Yes, yes. We do have a very good chemist on campus, Luke Levis. He engineered what kind of halotag ligand. Some of the fluorosis, they are like a fluorogenic. So they wouldn't be fluorescent to then bind to the tag. And we have a range of colors as well. I think the current limit is we don't have an orthogonal halotag. So halotag is the best tag, much superior than other tag like snap tag. I think the snap tag, because the k-ohm is about 1,000 times lower than halotag. So the concentration, if you want to achieve the same concentration of labeling, same labeling efficiency, you will need a much higher concentration of snap tag ligand, which will introduce a lot of background in the cells. So what we find is a halotag. People have tried to engineer orthogonal halotags, but it's appeared to be quite difficult to do. I think we at Janelia, we made enough attempts trying to do that. Yeah. Orthogonal halotag. So orthogonal halotag. So what they do is they try to engineer the pocket of the halotag binding sites. They try to engineer the pocket and see they can add also changes the synthetic ligand to see whether we can engineer a tag that is orthogonal, basically. Exactly. We can only follow only one at a time. Yeah, one at a time.
I guess, which is the big problem of genetic engineering or express.
Yeah. Yeah, that's true. Yeah. 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 And also it's very expensive if you buy from commercial source. But we have a chemist, they make the dye very cheap, and then we can do experiment. You can inject to animals and they feed flies. So we screen the different, and some of the ligand can actually go into brain as well when you feed through the food. But I do think, yeah, it's currently still have some limitations. If you really want to do the in vivo big animal studies, I think the cost is one of them. How you can do? I mean, you can email Luke and get enough dice and yeah, but
as opposed to giving the cell the apparatus to synthesize it.
yeah, exactly.
It's not plural. Right, sorry. The molecule added, which this configuration is present.
Yeah. And yeah, I think you any questions on halotag?
The wire.
You can, I think for larva, you probably can, yeah, put both for larva and the adult fly, you can put it in food. They actually can travel and then go to label the halotag. But I think for those type of scenarios, probably it's even harder to do imaging, right? Because the tissue have like scatterings, and so for you to observe single molecules is very, very challenging. Probably you have to use lattice light sheet, which, yeah. Okay, so that's good to know. I need to prepare what's a halotag next time. So the first question, very simple and a little bit of dumb question we're asking in the beginning is how does the transcription factor find its binding sites in live cells? So we have 2,600 different transcription factors, roughly 10% of genes in the human genome. And each of these transcription factor facing a daunting task, right? So it have to navigate through the complex 3D nuclear environment and search through 6 billion base pairs of DNA and eventually land onto a tiny piece of a target, and usually only consists of several DNA base pairs. And at that time, we are wondering how long it actually takes the transcription factor roughly to find the binding sites. I mean, you guys can take a guess, take days, hours, or just minutes. I mean, brief results here. I will come back to this point, I promise. Nobody? Okay, let's move on. So to really address this question, we decide to directly visualize how they move, how transcription factor move and diffuse in the cells. So at that time, we decide to use one of the called the multi-focus microscope. We first built in Moscow Stephenson's lab. And then we inherited this microscope in the transcription imaging consortium. After Marx passed away, very unfortunately. So a very unique feature of this microscope is we can simultaneously observe 9 different focal planes, roughly expand like a 4 micron range. So the Z depth for the cells we work on is roughly 5 micrometers. So this 4 micrometer depth allow us to cover the whole nucleus. And here we are moving the beads. You can see from the bottom to the top, and the light is basically goes through a multi-focus grading, and also from a chromatic correction grading, and then refocus the light from different focal planes to different place on the camera chip. So you can see the beads get refocused as we move the beads along right into different planes. So this microscope allows us to really simultaneously observe and molecules in 3D without the worry that some molecules will diffuse away in Z. So we, oh,
Like if you split up the 5 micron plane.
9 planes, yeah. So it's roughly 500 nanometer between planes, yeah. It's here we have a multi-focus grading. So I think they have these two-phase multi-focus grading have different patterns that can basically refract the light from different focal planes to the second for the chromatic correction grading. It's basically to correct, because there are some color differences. Each color have different, they will just align different colors. And eventually, we can also do simultaneous 2 colors as well if we split the light to 2 different camera chips. But of how this multi-focus grading is being designed, I think they run a lot of simulations. They try to improve the efficiency. In the beginning, the efficiency is about, I think, 80% with a 2-phase system. And eventually, they really try to push to like 8-phase and to get it to 95%. But the manufacture is really hard. I think they collaborated with with NASA. They couldn't make the correct design. So eventually we just stayed with this 2-phase system. But yeah, so yeah.
Just to show you the 9 planes that you on the left, and the pivot on the right is from the side.
The imaging from this, yeah, basically the imaging is showing you are moving. We rotate the image, so showing you what's happening in the Z direction. But the image is actually, we can rotate it back, so you will see the same, but we rotate like this way. So you see the beads is moving from the bottom to.
Yeah, this is. So if these are 9 XY planes, this is a XZ plane.
Yeah, exactly. Yeah.
And you project it, why some things that are bright, these happen to be lined up along the next.
Yeah, exactly. The imaging we basically limit by the camera frame rate. So in the beginning, we use the EMCCD. So the EMCCD is roughly, because we are doing full frame imaging, so it's roughly 30 frames per second. But yeah, if you use a CMOS camera, you can go as fast as you want. I mean, so I think that's the limit, is how fast the camera can go. So how much photon you can pump out from the molecules, because we are definitely splitting the light to 9 focal planes. So we need a really high laser power as well. So the observation window is very short. So observation window is 20 seconds, and we don't generally go longer than that because the cell probably will die. So in one shot, yeah. Huh? Oh yeah, we just want to show you, because if we take a single shot, it will be only showing up in 1 or 2 plane, right? If we take a single shot of the beads, we're only showing 1 or 2 planes. So the purpose of here is
The 9 planes that they image simultaneously,
the beads are moving. Yeah. The beads is a demonstration that we refocus them into different planes, right?
we make a stage go down.
Just to see exactly. Just to demonstrate that we indeed can refocus these beads at different focal depths. So this is just a demonstration. We don't move stage. Yeah, we don't move stage because here all the beads are in a single flat surface, right? So to convince you that we really can image 4 micron, because if we only look at one plane, the beads are only going to be focused, for example, in these two planes here.
You don't get
1 nice field of view, yeah. So we are looking at a single nuclei. Yeah, definitely. We are looking at a single nuclei. So this is another limitation of this technique. Yeah, the field of view is very small. Yeah. We just need to generate. So we basically combine this. We just crop out different regions, combine to a Z stack. For each frame we take, we have a Z stack of 9 planes. Then you have time points, M1, 1 to N. And then we can do just regular 3D localization on these time points. So you can basically track where those molecules are. We are using 100x in these experiments. Yeah. With 60x. I think it's specifically designed coupled with that like objective. Yeah. With the 100x. I think all the gradings is coupled to that like objective. Yeah. Yes. We probably need to redesign the multi-focus grading and stuff, and then we can potentially go to 60x as well. Yeah, I think that's Yeah. It's a low throughput technique in sense of the cells, but it's a high throughput in terms of molecules, right? We're tracking a lot of molecules simultaneously, but we are not looking at the like a variability between cells, right? So the variability between cells, we have to do many cells. So that's the limit. Yeah. So the numerical aperture is actually going down when you're using a big field of view. for example, 60X generally have lower numerical aperture. So for all the single particle imaging experiments, we actually prefer to use 100X, which have a 1.49 NA, so almost 1.5 NA like length. So we collect a lot of light, we can see the molecules clearly, especially in this case, because we're splitting light to 9 focal planes. So we need the signal to be collect as much light as possible, and we don't want, otherwise the molecule become too dim. Potentially, you can have a opposing objective. Yeah. If you have an opposing objective, but that's going to be much harder to align. If you work with microscope, you know the alignment is very, very tricky, right? And sometimes people just give up doing experiments because of the alignment problem. So, yeah. But yeah, I think that's as far as we went with the experiment. But the idea is we localize them in 3D and we link the molecules like the nearest neighbors through time. So we have a single molecule trajectory. And we try to keep the molecule very sparse, so we don't have crosstalks. For example, one molecule move very close by, and so we have these tracking arrows. So as you can see, we can track molecules in the cells, and I think the video is, we can track molecules in cells in 3D just to look at how they diffuse and how they bind. We generally acquired a lot of these single molecule trajectories, and just to figure out how the transcription factor do its job. So what we find is those transcription factors are not super smart. They basically use this try and error mechanism. So they bump into a place and then they diffuse, and then they diffuse around again, and they have different binding times. Generally we believe at a specific binding site, the binding time is slightly is longer than the non-specific binding sites. So when we really, based on our data, we calculated how long do it take if we only have one binding site, one transcription factor. It roughly takes that transcription factor about a month to find that sites. So the question, I think, how we still alive is to take a transcription factor a month to find a binding site, right? So I think the trick is the cell planes will have such a high copy number of transcription factors in the cells. So from the DNA perspective, basically we dividing that 1 month's time by, for the transcription factor we look at is roughly 100,000. So we divided that 1 month's time by the copy numbers. So if we calculate it that way, it's roughly the binding site being sampled every 20 seconds. And also another advantage, I think, evolutionarily, if we require really high copy numbers, and if we decrease the copy number very quickly, for example, we degraded the transcription factors, which will allow us to very rapidly tune the system, allow us to control the downstream processes. I think that's our one speculation why we need such a high concentration of transcription factors, makes the system more tunable. Which I'm not like a theoretical physicist. So if my statement is not correct, please, correct me anytime. Okay. So, and later on, we also, for attachment.
Between 2 DNA localizations.
Yeah.
Is 4 seconds.
Yes. Yeah.
And it stays 0.8 seconds on.
Yeah. I mean, it's roughly. I think we have a mean lifetime around amount.
So what did you use to distinguish between
the sliding, you mean? Yeah. The sliding part we the sliding motion we couldn't localize. We don't have localization precision to look at sliding on DNA. So I didn't go to details, but what we did is we attach DNA oligos to the glass surface, and we use different lengths of the DNA oligos, and then we introduce heterotag labeled SOX2 molecules. So generally, what we find is the longer the DNA molecules, the transcription factor actually stays there a longer time. So we infer is there some sequence length dependent, because the resin time is length dependent. We infer there probably some sliding there is going on. But in live cell experiments, we couldn't distinguish binding from sliding. Yeah, and yeah, I think this is a very controversial video. This is a combination of both, and this is a very artistic view. Yeah, 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 yeah.
Why? I think the issue is more than exactly like
we have some evidence I will show you later. But I do think it's a very wide range of like interaction time, and this point, don't take the 0.8 second very like literally. And we, based on one of the measurements we have, I will show you later. We have this number, but because I think the limitation of all the single molecule measurements, we are always selectively looking at one time windows. We couldn't capture all the full range events. How long? Yeah, 20 second-ish. Yeah. I will come back very quickly. Just bear me a couple of slides. And maybe I incorporate too much information. But later on, we also adapt this technique to look at protein-protein interactions in the cell. And here we are looking at Huntington. So the purple spots appeared here are Huntington, Y-type Huntington proteins. So they diffuse around in the cell. And then we also have a mutant Huntington protein form aggregates. So they have a polyQ stretch form aggregates in the cells. And as you can see, there are higher tendencies for the molecules, for the Y-type Huntington proteins to be captured at the surface. And we can also adapt these techniques, live cell palm experiments. We can accumulate localizations. We present in a rolling time window. So we can look at high resolution protein formation, protein condensate formation dynamics in live cells. And after these applications, we also, right? Look, how many GFP molecules? A lot. We didn't estimate, so it's a big aggregates. Single heterotag, yes. Yeah.
Very beautiful demonstration of photon yield.
Oh, they are acquired in different channels, right? So the laser power is different. And we try not to bleach the GFP because it's very easily get bleached. And we do use high laser power to observe single molecules. So I think.
But it's nice to see.
Yeah.
Much higher photon yield allows much more stringent localization.
Yes. I mean, if you want. Exactly. I think, yeah, we can, yeah, much better than GFP. If you want to do in cells, if you want to do in vivo, I think GFP still have advantage because you don't need to put the ligand. Yeah, but Thomas make a really good point. If you really want to set up easy demonstrations, I think HaloTag and yeah, the student should be able to see the molecules very well and under the microscope. And it's very exciting, but everybody see the first time is always very excited. I mean, after years, I'm slightly less excited now. Oh, so this is basically how many polyglutamine this Huntington protein have. The wild type Huntington protein have 25 glutamine. And then this mutant Huntington, they have this extended polyQ stretch. So it's actually the Huntington's disease is caused by this extended stretch of this polyglutamine. So, and then once they have this polyglutamine, this protein tends to form kind of aggregates in the cells. And when we frap those aggregates, they don't come back. So they're very stable, like hours. When we track for 2 hours, they don't come back. There is no fluorescent recovery. So which suggests they form very stable structures in the cell. Yeah.
So you can bleach this larger GFP.
Yeah. Oh yeah, this is, you can see the how many seconds, right? We only shoot 4 seconds of movies here. So in a 4 seconds, they don't move much, I think.
So you're saying that if you bleach the green,
they wouldn't repair after 2 hours. We wait for 2 hours. Yeah.
So
it's a lot of condensate. I think people study condensate, they find it when they frap it, it recover within seconds. So it's definitely not a very like phase separation. The dark.
once you bleach it, it's dark, but it's not necessarily color.
Oh, it's definitely not done. Yeah, it's there. It's just it's no exchange between the free floating molecules to the aggregates. Yeah, not like a lot of these phase separation. These people when they frap their condensate, it's come back within seconds. Yeah.
And why do you want to say
It's that we shouldn't say
that?
like aggregates dynamics. I think condensate people have maybe phase separated condensate, right?
Demonstrated.
They are not phase separated like aggregates. Yeah. I can change the title. I mean, not to avoid confusion. It's not a liquid phase condensate. Yeah.
But some point, like you go into condensate, solve that deep or some was then bypass.
I think it's because of the focal depths. My guess is we are actually covering some surface, right? Because the condensate like that, the aggregates look like that. We are looking at like this. So there are some aggregates probably shaped like that, and you see it go inside. Probably it's interacting with the surface because the focal depth is about 0.8, 800 nanometer, right? So we're looking at the 800 nanometer range. So if the condensate have aggregates have irregular shape, some surface might appear. So you actually interact with the surface there. Yeah. Not coming out.
Once they land on that, they stay there.
They get higher probability to get trapped there and then get bleached. And probably for the getting out part, I still think molecules can, I don't know what's the resin time here. Yeah. But I mean, which is a visual demonstration that it's have higher probability to get trapped there. Yeah, just look at the protein-protein interactions. Yeah. So later on, we also basically maybe I think I prepared the slides in a wrong style, but I should really focus on one or two stories. But so later on, we also devise these long-term single molecule imaging strategies. We can do very sparse labeling, and so we can track the vesicle proteins in neurons. So we discovered axon, basically the enhanced like anterograde trafficking dynamics of these synaptic vesicle precursors at the axon initial segment. And later on, we also adapt this technique to study how two differential sorting of two different sodium channels into different neuronal compartments. So this, and then after we publish our work, and I think these studies have generated a wide interest to use our techniques to look at different molecules, and we get a lot of requests from the field. And one of the requests from Jennifer Downer's lab to study this how the CRISPR-Cas9 molecules actually target the search in the live cells. And so probably I don't need to introduce too much about the CRISPR-Cas9. So basically, it have a guide on it combined to DNA in a sequence-dependent manner. We have an active Cas9 that can cut the DNA. We also have a deactivated like a version that only bind but does not cut. So here is a movie. Here I'm not a theoretical physicist. So we mainly like a visual data, so we can track basically these CRISPR-Cas9 molecules as they move inside the cells, and we can look at CRISPR-Cas9 trajectories, and we overlay them with a heterochromatin marker here. So there's a heterochromatin marker called HP1, which labels the heterochromatin in the cells, and clearly you can see that the CRISPR-Cas9 protein don't go inside of these heterochromatin that much. So they sample this heterochromatin much less compared to the sampling what's outside of the heterochromatin. So we also here just a broader demonstration that we use the single particle tracking techniques, and we look at the different molecules with our collaborators. And for example, we collaborate with Nat Hans lab at the Rockefeller, look at this MECP2 and different MECP2 mutant proteins, how they diffuse and how they bind to chromatin or bind to structures in the nucleus. And most recently, and we collaborate with Howard Chang Howard Chang and Paul Mitchell's lab to look at this extra chromosome DNA and look at the structure and the dynamics in cancer cells. I mean, just give you a brief, broad demonstration that this technique can be widely applied to different systems and reveal some insights into that system. Yeah.
That was with
This is with 2D particle tracking. Yeah.
also
Yes, this is 2D. Simultaneous imaging. Yeah, we have a setup with 3 cameras, so we can actually track 2 different structural markers on top of the single particle. Yeah. I think deep tissues problem is mainly limited by the objective, because the objective we use is 100x, like oil objective. So the spherical aberration is very high when you go too deep. So even like 5 micron into the cells, the quality of the single molecule will drop a lot. So I think if you really want to do that, I think it's probably the best is using silicon oil lens currently available with Nikon, and which with much less this spherical like aberration. And also we need some optical sectioning technique. So currently we're building a microscope that we couple spinning disk with really high laser power. And then so we can achieve optical sectioning, and then we can, in a relatively deep, probably 50 to 100 micron range. So yeah. Those tracks are how long? I don't have a timestamp here, so, but I roughly, I think we tracked 20 second, like 30, yeah, 20 second, like we could 20 second. Well, if we go in with a 10 millisecond, we will get 100 frames per second. So it's 2000 frames. Yeah, 2000 frames, 20 second. But we couldn't track molecules for the 20 seconds. Some of the molecules we are diffused away. So I think in general, we are selecting longer trajectories to look at, as trajectory at least have 5 like constitutively localized localizations. Yeah, 5. Yeah. Yeah, we basically filtered out shorter trajectories. Yeah. Yeah, because just for the like MSD analysis, we filtered out some of the shorter trajectories. Yeah. Average length, my guess is 10 frame, yeah, per molecule something. But we have longer, some is longer ones, and we have shorter ones. This is not multifocal. This is a single plane. This is just a single plane, because multifocus, the limitations, we don't have 2 color simultaneous. So in this experiment, we wanted to have 2 colors simultaneous. So we go with our other microscope. We have 3 cameras, and we allows us to look at the heterochromatin simultaneously with single particles moving around. Yeah. So we do lose a lot of molecules when they diffuse out of Z. But if we assume the diffusion is more or less XYZ is isotropic, so when you trap molecules in XY, it will reflect its movement in Z as well. But that's some assumption we made, and it's generally accepted in the field currently. A lot of people, they just do 2D tracking. Yeah. Okay. So a couple of observations we made at single molecule level really pushed our inquiry into this genome organization field. So I mean, I want to share these 2 very exciting observations with you, and also address some of the previous questions where the 0.8 second is being estimated, the non-specific binding time. So what we did is we use a really low laser power and long acquisition time, so allow us to blur those fast diffusing molecules in the cells. So if we blur those fast diffusing molecules, this allows us to more or less selectively look at those immobile events. It's just like you are looking at, you're taking a picture in a dark environment. If you wave your hand, you don't see your hand anymore. So when we counted how many, using this technique, when we counted how many frame each molecule stayed, and roughly we observe a 2 component. It's a short-lived component and a long-lived component. And if we delete the DNA binding domain, this long-lived component dramatically decreased, which suggests the reflects some sequence-dependent protein DNA interactions in the cell. And we followed up with this observation later on. So basically, we want to devise a strategy allow us to systematically map where this stable binding event occurs in the 3D space in the cells. So we use a bright GIF dyes and we use lattice light sheet imaging. And we basically do this iteration 3D scanning and we, yeah. Yeah.
So this is you're showing the correlation. So
it's a survival, I think this is survival. Oh, you mean in the image?
Well, so the thing that's plotted decay. What is decaying? What are you measuring?
Well, measuring how many molecules still survive after 10 seconds or after particular time.
Survive in the whole
in the whole, so we basically track the molecules, right? So, and we see when the molecules start to bind, we count. And then when they either bleached or leave, and we count that as a resin time.
You start counting them when they
appear.
When they stop, they appear.
Yeah.
Oh, I see. And they don't appear if they're
Exactly. Yeah. And then, so we generally believe that slightly longer these binding events are sequence dependent. So that's our assumption at that time. And then we want to map where this stable binding events occurs generally in the 3D space. So we use this halo tag, we control the sparse labeling and let the halo tag ligand diffuse very sparsely in the cells, and we do this iterative 3D imaging. So what we observed is very striking is this, when we accumulate, here are 7,000 stable binding sites we mapped in 2 different embryonic stem cells. And we observe this clustered pattern you can see. And here's the localization like densities. The hotter the color means there's more local neighbors. So which suggests clearly you can see those stable binding sites, they are not homogeneously distributed in the nucleus. Instead, they form these local density hotspots, what we call enhancer clusters at that time. And these results really lingered in my mind for a couple of years. I was always wondering what kind of DNA structure actually underlying are responsible for these clustered binding sites. And then I went to a meeting and I met Howard Chang. And at that time, they are developing this currently very popular genomic technique called ATAC-seq. So the idea here is they have a hyperactive transposes called TM5. The TM5 can insert DNA oligos to the accessible chromatin with high densities. Because the SOX2 only interact with, is mainly interact with the like open chromatin. So we, at that time, I was thinking maybe we should just map where these open chromatin are in the cells. So we put a dye onto the DNA oligo, and then the enzyme will insert this dye to these open chromatin sites. And then we coupled this with a 3D palm imaging using lattice light sheet. So here we also introduce the astigmatism, so allows them to more precisely localize the molecules in Z. So what this is we reconstruct image. So we see that once those are accessible chromatin sites, they also form these clusters, right? So it's mimicking these SOX2 enhancer clusters we are observed in live cells. So now instead of looking at these 2D maps, 2D high-C maps that generate from millions of cells, now we have a single cell point of view. What's this accessible chromatin structure generally look like? And we can also validate this by using DNA fish probes targeting these accessible chromatin domains. And you can see the roughly the overlap pretty well. So with this technique developed and validated, and so we decided to focus my lab's research to this very fascinating protein complex called the cohesin complex in the cell. So from the electron microscopy images, you can see this cohesin complex, they form this flexible ring-like structure, roughly with a diameter about 15 nanometer. And there's 3 main components. is this SMC3, SMC1, and also a linker protein called RAD21. And there is also an accessory protein called NIBL. So this NIBL can stimulate the ATPase activity in the SMC3. So once the ATPase activity is being stimulated, people propose that this ring can translocate along the chromatin to extrude the DNA to form these chromatin loops. And this loop formation have higher probabilities to be stopped at these CTCF boundary sites. So at that time, our question becomes very simple, right? So we observe these clusters and people propose cohesing are very important. So our question is, what happens if we get rid of cohesing? What is going to happen to these accessible chromatin clusters that we observed? So that's the experiment we're trying to go after next. And we did some simulation experiments with Bing Zhang's lab at MIT. So when doing simulation, if we only consider this loop extrusion model, and we ask Bing to predict what's going to happen to these accessible chromatin clusters. So based on the loop extrusion simulation, molecular simulation he performed, he believed that after cohesing depletion, we should observe much less clustering and fewer clusters in the cell. So we just want to test whether this is the case. So we actually perform the experiments, and we using a CRISPR-Cas9. So we knock in a halo tag with a diagram, a protein diagram. So we can add a auxin. So once we add the auxin in the cells, this RAD21, this linker protein, can be very rapidly degraded. So just within a couple of hours, 95% of the protein is gone. And then we couple this assay with 3D ATAC pump. So what we observe is completely opposite to the model prediction. We actually observe this much higher clustering of this accessible chromatin in the cells. And here just give you a single cell point of view. So when we deplete the cohesing in the cells, these accessible chromatin, they start to appear, they form these much larger and more connected structures in the cell. Keep a really long story short, so we perform a lot of genetic perturbation and also chemical screens. And eventually we identified a protein called BRD2. And this BRD2 protein it have a bromo domain that can bind to accessible chromatin, bind to the acetylated chromatin. So what happens if we get rid of the cohesing, the homotypic interactions between BRD2, you start to bridge and fuse this accessible chromatin site together. And this concept is also being validated when we work with Danny Tsai so a co-advised postdoc with Jennifer Lippincott-Schultz. So what Danny worked on a transcription condensate called the YOP. So this YOP protein can form these condensate structures very rapidly after hyperosmotic stress. So though they find this YOP condensate can also induce this more clustered like a distribution of accessible chromatin domains in the cell. See, we use hyperosmotic stress. So basically we extract water outside of the cell. It just push water out of the cell. So this protein start to form condensate within seconds, within 20 seconds also. Yeah. Yeah. I think normally they also form, normally the YOP is outside of the nucleus, not until you, mechanically stimulate them, they localize into the nucleus. Yeah. It's everywhere. It's everywhere. You mean whether the concentration of YAP increase in the nucleus?
Whether you see the is either when the concentration of GABA increases, or is it just
Whether the concentration increases. We don't have system currently to test that idea, how to very rapidly increase concentration of YAP. But I would assume so, because when you have water, you are basically decreasing the volume of the cell, right? So it's kind of increase the concentration of YAP. So in that sense, probably that's the case. We increase concentration, have a higher probability for YAP to form condensates. Yeah. And then later on, we also worked another lab at the Johns Hopkins. So they study this G quadruplex induced formation of another protein hop called the DAX. And what we find, this DAX protein hop can also aggregate these accessible common sites. So based on all the data we have currently, what we believe the function of the cohesin is really to separate this accessible chromatin and also counterbalance the weak interactions mediated by different proteins, just to prevent them spatial mixing in space. So the next question we ask is, what's the physiological function of these clusters we observed, right? So, yeah. Yes, we did. Yes, yes. Oh, I didn't show the data here, but we do have these autocorrelation function going down. Yes, yes. I should, yeah. So here we are basically dipping our toe to a really long-term debate in the field. Because for a really long time, people believe there must be a structural function relationship between gene organization and the transcription or regulation. However, this belief is being challenged in the past 7 years or so, because when people acute deplete cohesin or CTCF, they don't detect significant gene expression changes at the cell population level. And vice versa. So when people acutely inhibit the transcription activities, it also have very subtle effects on these high C mirrored domains and compartments. So those results start to accumulate and are casting doubt in people's mind. Maybe these two things are functionally decoupled. So my lab can repeat these experiments in my lab. So what we did is we acute deplete this linker protein RAD21, and we perform cell population based on RNA-seq. And indeed, we detect very subtle gene expression changes. However, which really doesn't compute with us is this dramatic change of this accessible chromatin in single cells, right? So this is just keeping in my mind for a really long time why we have such a big disconnect between the form and the function. So here I really would like to thank one of the brave postdocs in the lab, Pong. So he just started his lab at the Shenzhen Institute of Advanced Technology. So as you remember, one of the most dramatic observation we have is this spatial mixing of these accessible common domains. So we, instead of looking at the expression of a single gene in a population of cells, we start to ask what happened to gene expression correlations between different domains. If we rank the expression level of A and B at a single cell level, we can generate positive correlations, negative correlation, or no correlation. Well, the average level can stay the same, right? So we can, if we rank them, you can have these 3 distinct correlations without changing the average levels in the cell. So we really would like to test whether this is the case. So and just to elucidate, and when we doing cell population-based measurements of chromatin accessibility across different sites in the genome, we didn't observe significant differences. This is measured in bulk. And the question is really what happens to the correlations between domains, right? Between two accessible common domains. So, and we devised these experiments with single cell ATAC in collaboration with the Howard Chang's lab. And so so we want to measure the correlations, basically what's the chromatin co-opening probabilities in single cells. So when we analyze data this way, what we find very strikingly is when we deplete the RAD21, we can see this dramatically higher chromatin co-opening probabilities across all the chromosomes that we examine. Here, just showing each triangle along the diagonal line here is basically one chromosome. And here I'm just showing you what's happening on the chromosome 2 here. So this is the data we get. So the hypothesis we have at that time is what's the function of the cohesin in single cells is really to decorrelate gene expression in time. So if we have two genes that are really far away, and in the Y-type cells, they tend to not correlate in time. But after cohesin depletion, they tend to co-fluctuate in time. And because when you do bulk measurements, you are looking at the middle lines, right? So we like average many cells, basically we don't observe this phenomena. So also we look at the blue spots in the middle here. Not until you're doing correlations in single cells, you will start to observe this the covariance of these two genes actually change, right? So to really also to test this, we'll also go to single molecule type experiments. So we designed intron probes against genes that is very far away along the chromosome. So some of these genes can be like 26 million base pairs away. So when we in stem cells, when we deplete the cohesin, what we find is the co-activation frequency of these genes become higher, and also the physical distance between those genes also become the co-activated genes also become shorter. But if we look at the single genes across the population, we don't see dramatic down or up regulation of those genes, or at least a constant trend. So what we believe the function is really is not to determine the average expression level of single gene across the population, but to decorrelate those genes in time. So because I was trained as a biochemist when I was doing my PhD with TIJ, I have been exposed with a lot of these transcription factor complexes. So at that time, we also started to label these transcriptional complexes. So one of the transcription complex we label is this mediator complex. So it have a 25 subunit, roughly around 1 megadalton. And we labeled several of these components, and all of them form these clustered structures in the nucleus. So what we find is that after cohesin depletion, these clusters also become bigger in size. And here some caveats here. So we basically use these the same same thresholding. So the protein concentration doesn't change. We just use the same thresholding. So if the mediator become broader, it also will appear like a bigger. Did I lose most of you still can follow? Okay, so we can also do live cell imaging. We can see after cohesin depletion, there are some fusion events or spatial mixing of mediator hops is going on. So which consistent with the spatial mixing of accessible chromatin we observed in with the ATAC pump. Right here. This is a mediator 6. Color code is a volume. Is how big is the condensate? How big is the hop? Yeah, so it will become bigger in time. Seems.
So it's just labeled the GMP.
This is labeled with the emirate. So it's better than GFP. Yeah, it's slightly better than GFP. Yeah. We use the iris scan. So the iris scan we find it doesn't bleach cells that much. Yeah. Another technique we developed earlier is we can use this translational control, so we can basically very sparsely label any molecules in the cell. So we basically introduce a stop codon between the protein of interest and the tag, because there is a certain chance for the ribosome to skip the stop codon, and you can actually control that by using different like sequences. So we can really achieve super sparse labeling in the cells. We can control like 50 copy of the protein is actually with the tag in the cells. And this very sparse labeling allows us to very confidently introduce long dock times. So we are not worried there's some crosstalk, some molecules come to, so our tracking is get like messed up. So these are very sparse labeling strategy also allow us to look at this very like hundreds of seconds, this transcription factor behavior in the cells. And one of the behavior we observe is this very interesting, this local hopping events. You can see that one of the transcription, they stay still, and there is a transcription factor, they just locally hop around inside of the nucleus. So we also start to look at those hopping events after cohesin depletion. So what we find is that after cohesin depletion, these molecules tends to hop beyond longer ranges in the cells. In the nucleus. And so one of the hypotheses the reviewer suggests is maybe they are being trapped at some protein condensates, like the mediator hop, so they are they can only move within this range. But because the hop is become bigger, so the transcription factor can explore longer range. So based on these three separate studies, what we believe the cohesin connects the form and function in single cells. So what we find is the cohesin demix accessible chromatin. So without cohesin, the accessible tends to aggregates to form bigger clusters. And then also the function-wise, we believe the cohesin is to decorrelate gene expression in time. So basically, we can keep this gene more independently expressed. So I always been asked by the biologists, what's the physiological meaning of this? Why we need the cohesin in the cells, right? So why we need the cohesin system? And when I look at this question, I always come back to this co-accessibility map here. If we assume we have two genes that drive cells to two different cell types, and because if we in the Y type cells, we can put these two genes on the same chromosome or in a different chromosome. Probably doesn't matter that much because the regulatory landscape is relatively flat. However, if we put these two, after we don't have a cohesin, if we put these two genes on the same chromosome, they have a higher probability to get co-activated. And during the animal development, we need thousands of genes to be differentially expressed. But we only have 20 chromosomes in the mouse genome. So if you are going to run into this pigeonhole problem, right? You have much more pigeons than the host, so you are end up with more pigeons on the same chromosome. And here just give you another simulation experiment we performed. What we believe this function of cohesin is to probably establish this independent co-fluctuation domains on the chromosome. So with the cohesin activity get decreased, the more and more genes, they will start to co-fluctuate in time. And also one very interesting property of this system is that if we're looking at any cell population-based measurements, we are basically look at the middle lines, the black lines in the middle, because when we average from multiple cells, they are basically looking at the black line, which suggests any cell population-based technique would not have the resolution to tease this apart. And so I also believe this is probably why when we perturb a lot of these cohesin related molecules, it's really hard for us to find functional outcomes at the cell population level. The simulation is I'm sorry. So this is a chromosome. For example, this is a chromosome. And then this is the gene activity. And they are independently, with cohesin, they are independently fluctuating. Without cohesin, they become more and more co-fluctuating in time. Did I explain? Oh, this is more like elucidation. Yeah, like elucidation, what we believe, what's our hypothesis. There's not much underlying, principles or something. It's a very simple simulation that with a MATLAB code I write. Yeah. I mean, in my mind, this could be explaining why we couldn't see this functional outcomes of the cohesin depletion. And then also we can put this into mathematical terms, right? If we put genes encode different cell types along the chromosome in the Y-type cells, because they are independent, so we can, these probabilities add up to 1. But if we get rid of the cohesin, basically we are introducing this probability are not independent anymore. So we are introducing a probability of confusion here. So which suggests the cells will lose certain potential to differentiate to all the cell types. And you might ask whether this is actually underlying any human genetic like diseases. And indeed, there is a human genetic disease called Collinean-DeLange syndrome. And when you look at the genetic pathways, almost all the mutations occurs in a single genetic pathway. It either occurs on the protein that responsible for loading the chromosome, loading the cohesin ring to the chromatin, or on the cohesin ring itself. And this is a very weird like disease because the penetrance is highly variable between different individuals. Some kids have problem in the brain, some kids have problem in the GI tract, some have in the limb. So it's affecting all the like organs, and every individual is different. So based on our observation, what we believe is that possible this random gene co-activation or stochastic gene co-activation during the development, because they probably random co-activate certain genes, right, to drive like a downstream, like a deterministic effect for affecting this individual, and some other genes is affecting other like tissues. So that's our hypothesis currently. So the challenge is how do we, if we want to test this hypothesis, how do we get to measure the co-activation of thousands of genes in single cells? So here we are. So that's why my lab is also start to doing this spatial genomics experiments. And it's also single molecule based. I will try to explain this. And I think it's also based on what Eric proposed. So if we have many molecules in a diffraction limited spots, it's like RNA, DNA. Instead of separate those molecules in time or in color, we can separate them by like a hybridization rounds. So we can use sequence-specific probes, go inside and label one specifically, and we can localize where they are. Because the sequence specificity, we can also tell what they are, right? So we can know the identity of the molecules, we know where they are localized. But one downside of this technique is the throughput is very low, right? If you want 1,000 target, you need to do 1,000 hybridization runs. So I think the true innovation in this field in the past, 10 years is probably this, how to barcode them, right? How to barcode these, to compress this into less like hybridization rounds. So to really test our idea, whether this co-fluctuation actually occurs, so we designed intron probes targeting 208 genes across chromosome 2. And then we can encode these 208 genes just by using 12 hybridization rounds. And here we can zoom in and look at the gene bursting events of these 208 genes. And each gene is color coded. And so we can start to look at what's the co-bursting frequency of these 208 genes. So what we find is that when you look at, if we deplete cohesin, when you look at bursting frequencies in the population wide, they don't change that much. But if we start to look at co-bursting frequencies, the co-bursting frequency actually become higher, and also the physical distance between the genes become shorter.
What's bursting here?
Bursting just count whether we detect that genes with the intron probes and where they are. If we calculate what's the pairwise, or what's the frequency of the pairwise, the co-localization, coexistence in the cells. What's a fraction of the co-bursting fractions in the cells? Yeah. Yes, because the expression will more look at the nascent RNA production instead of looking at the steady state, like RNA here. So this is consistent with our model that if you get rid of cohesin, more and more genes start to co-fluctuate in time, right? So, so currently, yeah, this new observation really drives us to build the next generation spatial genomic spatial tools for deep tissue imaging. So that's currently my lab is fully focused on all the people, including me. And so I don't have more data to show because of the pattern problem. So I think, but yeah, that's surprising. Okay, but the idea is we're going to look at these relative gene expression correlations. I mean, I'm a little bit against looking at the early development because I think the cell cycle is very complicated. And when we try to tease out the gene organization. So currently, my lab interest is more in the adult tissues because they don't divide anymore. So we don't need to worry about which cell cycle phase they are in. And so we're going to study how the perturbing some of the factors affect the gene expression correlations in single cells. So this is acknowledgement for the people in my lab, and also some of the early postdocs that their hard work really pushed my lab to where we are now, and a lot of collaborators both inside and outside of the campus. And yeah, I think I already take a lot of questions. So should I? I'm glad it's the first time I do a 90 minutes talk. So and I'm survived exactly. Yeah. Thank you. Yeah.
It's my great pleasure to introduce Untitled Chapter, who is a professor of physics at the University of Maryland and the co-chair of the biophysics program. She done her PhD at the Notre Dame and then went on to work postdoc at the MIT and did a short stint at the UNC Chapel Hill before becoming a professor at the University of Maryland. And you probably know her very well because she's very active in the area of biophysics and quantitative imaging, and her work spans from areas from the immune activation and single molecule fractions to transcription factor dynamics. And especially in the last few years, a few papers have come out from her lab that are very exciting transcription factor dynamics, and we're going to hear about it today. So thank you for coming.
Great. Thanks very much, Miloš, for the nice introduction. And I must say that we were fortunate at the UMB Biophysics program to recruit Miloš a number of years ago. And also the students here, I am co-director of the biophysics program. So students and faculty, if they're interested, undergrad students, let them know about the program. I also want to thank the organizers for inviting me to this symposium. It's really wonderful to be here, and it was great to hear the wonderful talk by Shay. So I have been at the University of Maryland for a number of years, primarily at the Department of Physics, but also as a joint appointment in the Institute for Physical Science and Technology. And so a fun fact about me, as Miloš introduced, we do a lot of imaging on various different systems, but I also, as part of my passion or obsession, do photography. So I wanted to share with you two of my favorite images of New York City, since we are in New York. So I like to look at things at the microscopic and nanoscopic level in cells, but also like the sort of larger scale urban landscapes around us. Anyway, so today I'll talk to you about some of our more recent work on transcription factor dynamics, and all of that work was done in collaboration with Gordon Hager, with whom we are still working. But I'll give you a few introductory slides about some of our other work to give you a general sort of a broad overview of what we work on. So we're interested in studying cells at different scales, and my goal for a number of years has been sort of thinking about cells as physical objects. My PhD was in physics, always in biophysics, but what are the physical properties of cells, and how do these cellular components interact with the external sort of mechanical environment that the cell is in to eventually drive cellular function. So we're looking at it at multiple different scales, but I can't. Okay, good, thank you. Yeah, we're looking at it at multiple different scales. So for example, at the molecular scale, we have receptors, and as they go through receptor-ligand interactions, leads to enzymatic activity, clustering of receptors such as in immune cells. And then these sort of signaling events at the molecular scale can lead to larger sort of mesoscale dynamics within the cell. One of our favorite things to study is the cytoskeleton. And what you see over here is the dynamics of actin filaments, which go through these retrograde flows at the periphery and form intricate myosin-based cables. This is the synapse of an immune cell. And in that context, we're interested in looking at cell-cell junctions. So this is a T cell, and you can see the polarization of the T cell as it contacts a cancer cell. So you have large-scale organization of the cytoskeleton, the cell membrane, and various other events going on to facilitate the signal transduction across this junction, which is the immune synapse. And we've also on and off been looking at larger scale movements, for example, as you can see in this collective behavior of neural crest cells from chick embryo as they go through this epithelial to mesenchymal transfer. So you can see there's large scale sort of cell-cell interactions, but these interactions are also being mediated by the surface that the cells are in. And this is important in tissue organization and development. So these span multiple length scales and time scales. And one of the questions we've been asking are, how does the cytoskeletal dynamics and contractility, for example, drive changes in shape, receptor signaling, and function in immune cells, but also getting more and more interested in how do cells sense and transmit information, both chemical and mechanical, into functional outputs. So this is sort of the two broad areas in my lab. And so how mechanical environment and cytoskeletal regulate the immune response. And this was started in collaboration with Larry Samuelson at NIH. And then there's a number of collaborators that I'm currently working on this with. And then the transcription factor dynamics in gene expression in collaboration with Gordon Hager. So just a few brief slides about the immune response, because we spent a lot of years sort of imaging these. So I've just shown you an overview of the different types of dynamics we observed. You typically see these large-scale actomyosin organization. You have actin flows and myosin-based arcs. at the immune synapse when the cell gets activated. And this depletion zone of actin is also important for amplification of the signal at the center of the synapse, but also in the function of cytotoxic T lymphocytes, for example, because they secrete granules through this depleted zone. Microtubules are also highly dynamic. As you can see over here, these are the tips of microtubules moving across the synapse. And in this movie, you see sort of large-scale shape dynamics of microtubules. And in some of our, yeah. Okay, sorry, I should have said this is a biomimetic. So this is a model immune synapse. So what we do is we can look at cell-cell contacts too, but to really visualize that high resolution, we mimic the target cell with a planar surface that is coated with the antibodies or antigen that the target cell will present. So in some ways, it's sort of a mimic of an immune synapse in that sense. Yeah. Is the synapse. And actually, we are looking up because all the imaging we do is inverted microscopy. Yeah, absolutely. I didn't take time to explain this at all, but that's true. And what we found is that there is a strong sort of interplay between actomyosin organization dynamics as well as the microtubule cytoskeleton in these cells. But also these dynamics are important for generating forces in these. And what you see again here is now traction forces being generated by T cells at the immune synapse. In this case, instead of a glass or other surface, we have a thin gel on top of which you have the cells spreading out. And then by looking at movement of beads embedded in the gel, we can measure the traction forces generated by the T cells on these surfaces. And it's been found by us and many other people working in this field that forces are important both for signaling activation and in regulation of how cytotoxic T cells kill their target cancer cells. And we're working on this also in the context of CAR T cells more recently to help understand better approaches for immunotherapies. We've been interested in work sort of in different types of mechanical cues, such as stiffness, topography, or mobility of the ligand itself, in how these shape cytoskeletal dynamics and signaling in immune cells. So that's sort of a more of the larger scale question. So we also can see that these cytoskeletal dynamics can regulate immune receptor dynamics at the molecular scale. And so again, so these are just sort of very, this movie is not playing, just a broad overview to give you an idea, but what you would have seen over here is B cell receptor molecules sort of moving around as single molecules, very similar to the movies that Jay showed and other movies that I will show later. And we can get this tracks, we track these molecules and we get get molecules, trajectories with wide distribution of diffusion coefficients. And we can use perturbation expectation maximization, machine learning algorithm to extract a set of different diffusive states that best describe this sort of diffusivity distribution of these large number of trajectories. And I'll talk about this in more detail later. But the bottom line is that these molecules, just on the basis of their mobility, can be classified into these distinct states. And we find that signaling molecules occupy lower mobility states, and also that the actin dynamics by actin regulatory protein, such as WASP, leads to enhanced signaling and reduced mobility. So the bottom line here is that the biochemical interaction can lead to different diffusive or mobility states, and that these give us a window into the molecular interactions as well as the underlying microenvironment. So we then actually will be using this method to study some of the other dynamics that I will talk to you about. And just very briefly, we spent some time trying to understand how cells sense and respond, for example, to the stiffness of the antigen presenting surface. We found, as you can see, the cell-cell contact is actually a cell making, T cell, let's say, making a contact with the target cell, which is soft and deformable, not smooth and stiff. And so the forces that cells generate change and increase as a function of stiffness. We also found that signaling is modulated by stiffness. And we and others have shown that the cytotoxic function is also stiffness dependent, as well as many other attributes such as gene expression, for example, and cell fate determination. And these are some fun movies showing that the topography of the interface that the cells contact is also important to modulate their cytoskeletal dynamics that I'm showing here, but also the distribution of signaling, the calcium signaling frequency, and many other attributes. So overall, we think that the coordination of biochemical signaling, the forces that are generated by the cells, as well as the mechanical cues that cells experience will drive their function. So with that, I wanted to talk about, just spend a minute or so telling you of what we've been thinking about. So these mechanical cues not only modulate these dynamics such as cytoskeleton, signaling, forces, but have been shown over the last 2 decades to have profound influence on gene expression, which is important in development, cancer metastases, and also the immune response. So we've been getting more and more interested in looking at the nucleus. So all of the movies I showed you so far were on this 2D plane. We were looking at the cell from the bottom. So here now it's the same T cell that we have taken confocal images of XZ, like XY, multiple XY slices through the cell. And then what we're showing here is an XZ projection. So you can see the actin cytoskeleton in red and then the nucleus in blue. And you see that during activation, the cell sort of spreads out and the nucleus undergoes large-scale deformations. And one of our interests in the upcoming future years is to also try and understand how these nuclear dynamics change as a function of T cell activation, differentiation into effector cells, and how this modulates sort of the internal dynamics and organization of the nucleus. And sort of longer term, we're interested in how mechanical information may be transmitted to modulate gene expression itself. So in order to do the, so before that, and this is just giving you some background that there's been many studies. I just cite like 3 examples over here. The forces that cells exert, of course, are important during development. The stiffness of the extracellular matrix can modulate cell shape, migration, proliferation, but also cell fate. So the cell phenotype itself changes as you allow cells to grow on softer or stiffer microenvironments. And the same can be said for topography. It influences cells in many ways. And this is just one example that shows that it also induces modifications in histones inside the cell nucleus. So in order to get a better understanding of how all of this happens, yeah. Good question. But I don't fully recall in this particular paper which histone they were looking at. Often people, like which marker this is, I don't recall, but I can, this was not our lab. So actually none of these are lab. Yeah, exactly, yeah. But often people look at markers such as H3K27, ME3, the heterochromatin markers, or H2B itself, or some acetylation markers. But yeah, I should go back and. So, so Jay already gave a really good and expansive talk about things that are happening in the nucleus. So some of this might be repetitive, but I'll just go over it because I have 90 minutes. So as you all know, the nucleus is highly organized across several length scales, starting from nucleosomes around which the chromatin wraps around. These then form slightly larger scale loops and clusters, which are then thought to associated in these in these topologically associated chromatin domains or compartments, which then, aggregate into chromosomes. So all of this various different organization across different length scale and all the changes in gene expression that must be going on has to work within this sort of highly complex and hierarchical sort of multi-scale organization, because as you know, there's, the DNA is like meter long. It has to be packed into a few micron scale nucleus. So it's a very complex sort of packed environment. Also, gene expression itself is highly regulated. Some of the key players are transcription factors, which act as switches turning genes on and off. They bind to specific DNA sequences and then regulate gene expression, but they don't do so on their own. They work together with sort of these large-scale transcription initiation complex, which has many different cofactors, mediators. They have to bind to enhance the promoter sequences, which all of them come together and start the initiation of RNA polymerase II, which then leads to start of transcription and then eventually production of mRNA. So the other important thing about transcription factors is they can also relay information about external stimuli to mediate gene expression. And among the factors that we'll be looking at are steroid hormone receptors. For example, this is GR, glucocorticoid receptor. So when they are activated with their ligand or hormone, then they basically become activated and then they act as transcription factors. So they can relay information about the external stimuli. And so, of course, the question that is in everyone's mind is how do these interact with DNA organized in the genomic chromatin? So a lot of studies have been done, and in sort of very powerful are some of these population level studies, such as ATAC-seq to study chromatin accessibility, RNA-seq to study gene expression, and also ChIP-seq to study specific binding. And this has provided us with a lot of powerful information. I think this is absolutely important going forward as well. But at the end of it, it yields a static picture of the transcription factor chromatin interactions and also population level picture, sort of a population average picture of what's going on when you have a large number of cells. But a number of years ago, it was found, and this is one of the pioneering works from Gordon's lab, where they found that GR, the glucocorticoid receptor, is highly dynamic. And they did these FRAP studies, photobleaching recovery after fluorescence recovery after photobleaching, and showed that in this particular array that they had created, which allows 200 copies of GR to bind to it. It basically, so initially you'll see the fluorescence signal and then they bleach it. And then they see that within seconds, sort of 10, on the order of 10 seconds, it recovers. So it shows that transcription factor interactions with DNA are highly dynamic. And one must understand these dynamics to get a better understanding of how these then modulate gene regulation, gene expression. So some of the questions we've been interested in asking as part of a collaboration that started with Gordon Hager. This was part of this NCI-UMD partnership we have. And I met Gordon fairly serendipitously because he showed some movies that they were collecting in their lab within the nucleus, and then we were working on T cells. And so we started talking about how we could work together to better understand these things. But that has been a really nice collaboration with two graduate students, former graduate students, David and Kostav. And Kostav is currently a postdoc in Gordon's lab. And we wanted to think about what are the dynamic signatures of chromatin over timescales that are transcriptionally relevant. How do transcription factors interact with chromatin? And then how is it related to gene expression? And also the question that I had started out with is how do properties of the extracellular environment affect the dynamics of TFs and gene expression? So we've actually been working in the last few years on, the paths sometimes lead away from the path that you initially started on. And then hopefully we'll come back to the path that we started on. That's asking how do properties of the extracellular matrix affect TF dynamics. So in Gordon's lab, they've been working, he's been one of the pioneers in studying nuclear receptors, specifically the steroid hormone receptors, glucocorticoid, estrogen, androgen, and progesterone. These are inducible transcription factors. They're activated by a hormone that you just add to the cells. So GR, AR, and PR, they are actually predominantly cytoplasmic, and upon ligand binding, they translocate to the nucleus. ER is primarily nuclear. So here you can just see sort of this movie that shows on the order of about 20 minutes upon addition of the ligand, GR translocated to the nucleus. And once they get activated, they go and bind their specific response elements, either as dimers, but GR is supposed to bind as a tetramer. Yeah. Yes. So these, yeah, they go into the nucleus. So they're initially in the cytoplasm. Oh, the hormone itself crosses the cell membrane. Yeah, yeah. So in the sense that you can just add it to the media and then it just goes into the cell. I believe that's what happens. And these I'll show you later, but you've heard about these dexamethasone, which has been used a lot lately, a corticosteroid, and then estrogen, for example, for ER receptors. Yeah, that's what happens.
There's something about the moment, something to get internalized because there is a service receptor that takes them off, right? But in this case, they're just really confusing.
Yeah. Thanks.
So the hormone is the cofactor.
Sorry.
The hormone itself is not a transcription factor.
No, the hormone actually activates the transcription factor by binding to it at one of the domains. So there's a ligand binding domain. So the hormone binds to it and then activates it. So activates GR. So then it can behave like a transcription. I don't know. Does anyone know? Are hormones
Kind of like deceiving naming receptors because most people, biologists, who focus on the receptors will think about something.
And that was my problem too for a beginning, because we always talk about T cell receptor, B cell receptor on the surface.
In this case, they're actually inside the cells.
They're inside the cell and then they go inside the nucleus. They're not membrane receptors in that sense, but maybe they're called receptors because they can get activated by ligand. I think that's why they're called receptors, because for the immune receptors, for example, also they have to bind an antigen to change their conformation that allows it then to recruit signaling molecules. But I don't know if hormones themselves can do transcription.
But a hormone is a protein.
A hormone is a protein? It's a molecule, right? Yeah. Like estrogen, E2, yeah.
Yeah, it's to be made from some polypeptide. No, it's like, I mean, think about neurotransmitters, right? They're small, they're what biologists would call small molecule.
Right. Okay. So these hormone receptors then are, they work as good probes to study transcription factor kinetics and dynamics. They're very modular. There's a ligand binding domain, as I mentioned. There's a DNA binding domain that binds the specific DNA sequence. And then often they have this N-terminal domain, which is unstructured, made of IDRs. So the hormones that activate GR are, as I mentioned, DEX and CORD. And GR itself is important in various physiological functions, development, metabolism, also the immune response. And So the first question we set out to look at is what are the dynamics of these as they bind and mine from DNA, and how do different domains of GR and other transcription factors mediate interactions with chromatin, and how do these interactions then determine GR dynamics in turn. So for that, Gordon's lab has been working on single molecule tracking of transcription factor dynamics, and Jay already told you many things about it, but I will just give you our synopsis of this. There's a protein of interest that is tagged with a halo tag, and then you can get a halo tag ligand, which is fluorescently labeled molecule. So in our case, we use JF549 or more recently JF650, which indicates the fluorescence. The fluorescence of these molecules, the wavelength, and you introduce the tag protein by transfection or making a stable cell line, and then you wash in you incubate it with the fluorophore, and it's just a short incubation, 20 minutes or something like that, and it gets into the nucleus, binds to the halo tag, and then you extensively wash it to basically get a very sparse density, which is needed. And we're using this technique called highly inclined and laminated optical sheet microscopy, which is a type of turf, basically turf, but not exactly at the turf angle. So it creates an incline, so this thin sheet of light that goes through the cell and allows one to look through the nucleus. And with that, you are able to visualize dynamics within the nucleus at the single molecule level. So this is basically sort of the imaging workflow. So and then you can track these molecules and get their XY coordinates. And there's sort of two types of things one can look at. So sort of temporal, how long does a transcription factor stop, right? Before it unbinds, and then how much does it move in space, sort of XY. So we'll first talk about the binding times itself, and this is what a typical image of a nucleus along with the tracks will look like. And what we'll do first from these is to obtain the residence times or dwell times, because these can carry information about how transcription factors interact with chromatin. And the workflow typically is to look at all the bound molecules that we get and think about something like a chymograph. So I'm kind of unpacking a little bit what Jay already told you. If you did have to make chymographs of them, you'll see these streaks, and the length of one of these chymographs will tell you how long that molecule was bound and at a certain spot. So you basically get a bunch of lengths. And then when you, once you have these different sort of dwell times, you can get distributions of the dwell times from these time scales that you obtain from single molecule movies. So I'll talk about first sort of a conventional interpretation of SMT data that had been, that had been believed for a number of years. So, you have a movie like this, and this is sort of a frequency distribution of the dwell times that you might get. So how can you extract some sort of quantitative information about it? And just to point out here, something that I missed, the time scale of acquisition is very important. In this case, what I'm showing you is, this is just the amount of hormone, but imaging these at somewhat slow time scales, so 200 millisecond intervals, what we call the slow SMT. This allows us to also get longer tracks because there'll be less photobleaching. And so typically what had been done is to fit these distributions, the survival distribution, which is basically 1 minus the CDF of these dwell times. So we can identify when they disappear. So from the time that we start observing. So basically we can
define these dwell times, I have to tell you when it stopped and when it escaped. Right?
So when it stopped is when you, so it's basically you, so the only time you have is when you start imaging or when a particle comes into your frame.
Sorry. Not asking my question. If I walk one particle,
Right.
maybe you go back. If I walk one particle, so that we're, I have this diffusive projection, and then I have a little, I then have very restricted spatial motion, and then I escape, and I have another diffusive projection. Is segmenting the tracks into those bound and unbound parts, is that challenging or is that very easy?
So we're in the limit of looking at sparsely labeled molecules. So we just look at the molecules while they're in one track, basically, rather than, so I don't believe you're able to look at molecules that go away from a spot and then maybe come back to it. So
you basically, maybe it's a similar idea to the last one, blur things a little bit. Then you don't see anything when it stops, right?
yes, so if you take so if your time scale of acquisition is very long, then you won't be able to visualize the molecules that are moving faster and that may be coming in and out.
Okay.
Right. So it depends on the time scale of observation.
Slowly back forward.
For this particular thing, but we actually do multiple types of imaging on the same system. And very briefly, because I can't remember whether I put this in very carefully. If you image really fast, let's say with 10 millisecond interval, then what you can get are all the diffusive molecules that are diffusing around. But then if you image slower, like I'm showing, then you get the molecules that are bound in that sense. So with the slow imaging, we are missing most of the diffusive molecule.
Thank you.
Okay.
Our distinction and the cross what the distinction whether you are having a diffusive or non-diffusive.
How do you distinguish? So typically what we do is do the fast imaging to get a sense of the fraction of what are the bound molecules and unbound, okay, with the fast imaging. So that's with the tracking, and I can tell you more about the tracking. Is that your question? How do we distinguish them?
Well, clearly also still have really diffusive molecules.
We get a few. Yeah.
So how do you distinguish those from tracks that you just you're tracking the distribution part and some
So-
of them must.
Right. So that's why so I'm not talking about the spatial right now, right? So we're talking about how much they stop. So in that case, we use a bound. The ones that don't move more than a typical histone moves, those are the ones we consider bound for a dwell time analysis, if that's your question. So we use histones.
And also
I mean, we're not actually constructing the chymograph, we're just looking at how long the molecule stayed. We're doing single particle tracking. The chymograph is more a visual aid. Sorry, but what was your question? So we're looking at how long a molecule stays before it disappears. Right? Yes. Sorry, I'm not understanding your question. Yeah, this is just an example chymograph. Right? Yeah, so it's like if you had to take a line through the image. We don't do a chymograph. This is very illustrative. Sorry, I didn't realize this would lead to. We take the 2D image of tracks, and then we have X, Y coordinates over time, And then we use that to see how long a particle stayed. We're not doing chymography. This one? This is just, so if you have an image like this one, if you took a line through it and then projected it over time. So this is, sorry, I didn't realize this would be confusing. It was meant to make it simpler, and maybe I should remove it for my next talk. We actually don't make a chymograph. Does that make sense? Right.
How do we feature that into the quantitative structure of this?
So, I mean, the goal is to have a to have the light sheet not vary across experiments. So to have the same imaging conditions. But there may be some
And it doesn't work if you want to make real use of actual numbers that you get. If you get a number for one condition, you get a number for another condition. But then that number, de facto, the absolute value of that number doesn't have much meaning because it must depend on the sheet.
So maybe I should continue and we can discuss this. Maybe it might become more clear once I talk about some of the workflow. Yeah. But I think my answer to that question is that okay, so I'll continue. So how do we get these times? So, and I think the answer, part of the answer to, okay, so to come back to it, so we fit, right. So in people in the field fitted typically this to 2 exponentials and considered that this represented 2 types of binding, sort of short-lived, the smaller exponent, the smaller times constant for the exponent that were short-lived related to non-specific binding, while the long-lived molecules were thought to be representing specific binding. And this I'm just showing from a recent review showing that many different transcription factors appeared to show sort of very short non-specific binding. I think as Jay also pointed out, less than a second and then tens of seconds of specific binding. But when we started doing these experiments, and okay, so this just very quickly goes over why this makes sense. So the idea is that for specific binding, you'll have sort of very strong association with the specific DNA sequences that is going to lead to these long-lived states. And you might, then the transcription factors that are non-specifically bound might be undergoing hopping, sliding, and various other search mechanisms that might lead to longer-lived sort of dwell times. And then if you put it together in a simple model, you basically get that the dwell times can be considered to be bi-exponential. But as we started taking data and looking at the data more closely, we see that the double exponential fit also doesn't quite fit the data because there are large deviations of the tail. And then some other studies from the Gebhardt lab also suggested additional components beyond just sort of the 2 long-lived, short-lived. So from here, we realized that the photo bleaching correction must be critical. And this is also leading to the point that you were asking. So how do we get actual numbers from the movies that we get from the dwell time distributions? And so photo bleaching correction is essential. And how do you correct for the photo bleaching? So one can use histones as a standard for photo bleaching because they represent long-lived binding. They stay on the chromatin for a long time. And typically it's limited by fluorophore bleaching, the longest time scales that we can observe. So we did a number of trials. I'll just give you a short review of the photo bleaching correction method we think works well. So the idea is to image transcription factors at different acquisition times, sort of different acquisition windows, intervals, as well as exposure time to induce different degrees of photo bleaching, and then use image histones with the same acquisition conditions as transcription factors. And then we corrected for photo bleaching using a number of different methods that had been used also in the literature. And what we found for the previously used methods, even if we did the photo bleaching correction, the survival distribution of the transcription factors depended on the acquisition time. So then the photo bleaching correction was not correctly being done. So what we have been doing is to fit the histone survival to a triple exponential function and then use the slowest time scale as the photo bleaching correction time scale. And then correct the transcription factor survival with this photo bleaching time scale. So what I'm showing here is one of the methods that didn't work too well, where you corrected by the entire histone distribution or the exponential function. But when we correct it by the slowest time scale, then we see that all of the transcription factor distributions that were taken at different acquisition conditions fall on top of each other. So this is the photo bleaching correction that we've been using. And we think that this leads to at least a better method of photo bleaching correction than was done earlier. Is it perfect? I don't know. Yeah. Yeah. Typically, the slowest is tens of seconds or 100. Yeah. I'm not remembering the exact number. And then the other ones are much smaller. But also the number of and sort of that's the slowest one is the dominant time scale. Yeah. Yes, it's the same fluorophore. Yeah.
So basically, get the rate by insisting that these things fall on top of each other.
These three?
The three.
Here.
So say that if I measure at different acquisition rates, then I have different sensitivity for the bleaching.
Mm-hmm.
But there's some underlying thing that's the same in all cases. And so your model of photo bleaching would be selected by insisting that once you correct, the three things fall on top of each other, even without it.
Oh, I see. I haven't thought about that aspect, but that's I'll think about it, and maybe we can talk later. Yeah. Right. Yeah. But the problem, but the problem with that is for all our experiments, we don't do all these different things. So now that once establishing this, we do histone for every single condition that we're doing to establish the photo bleaching correction properly. But I see what you're saying. That would require us to do, yes. But that's a great idea. Okay, I will take you up on that. Because there is a system we want to study that we don't have a halo tag H2B for. So maybe we could do that. Yeah. We also did laser powers. Yeah. We did laser powers as well. Yeah. Okay. So I don't know if, Thomas, if that answered a little bit your question as to being able to do the photo bleaching correction with histones to then normalize to get information from transcription factors. But I can. We can talk. Okay. Okay, I don't, yeah, we can talk. Okay, so once we establish this, what you also notice, and as we noticed, is that the bi-exponential model was just not fitting very well. And this is just an example showing you a bi-exponential model with the photo bleaching corrected data. And in comparison, a power law fit to the data. So with this new photo bleaching correction, we saw that GR in this case activated by court shows a power law behavior. And this then suggests, instead of two characteristic timescales, short and long, that there's a broad distribution of timescale and hence binding affinities. There could be a lot of questions about this, so I don't know whether I should preempt it, but I will not. So of course, the caveat here is that we have a couple of decades of scaling at best. So what we would want to do in future with better dyes and better microscopes is to actually be able to collect data for longer and longer times to see, does this indeed hold over even longer time scales? Yes. Yes. Next slide. Yeah, so this is GR. So we did look at many transcription factors that Gordon's lab has been working with. So Halo ER, as you can see over here, Halo FOXA, Halo GR, they all appear to show power law distribution, but also some other transcriptional co-regulators and cofactors, CTCF, SMARCA4, they also show power law behavior. And CDCF, which is a structural protein, it seems to be very, very long-lived. So all of this is pointing to the fact that at least some transcription factors we looked at show this power law behavior. I don't think I have the citation for it, but another work using exogenous transcription factor in mammalian cells, the first author is Normano. They also found a power law behavior of these dynamics. And again, this suggests that there is this broad distribution of timescales. So this was actually somewhat disappointing for us because we really wanted to use the characteristic timescale to then see how external perturbations affect that. So in a sense, it was initially a setback, but I think we need to think about what this means for the biology. I think the exponent of the power law is still very meaningful because the shallower exponent point to greater, sort of more stronger binding, for example, the shallower exponents, and the longer exponents are steeper. The steeper ones have shorter binding times. And I don't think I have that data here, but for example, yeah. So we've done some of that, and I'm trying to remember whether I have it in my talk. But at least, for example, when we washed out the hormone after GR activation, DEX or COT, you don't get a complete washout because they're still pretty strongly bound, but you see a strong decrease in the exponent. Yeah. The long-lived ones are the ones that are more indicative of specific binding. Exactly. So it then appears that even non-specific binding does have, it's not just one thing. It's because, and I'll come to why we think that is happening. But that's sort of the picture that we are getting now. And just to show a cartoon, this picture of specific, very well-separated specific and non-specific binding affinities doesn't seem to hold very well because it would lead to a bi-exponential distribution. And so how can we think about it? So this is a very simple sort of cartoon-like model. And the idea over here is that there is a sort of a circle. If you think about the DNA as a circular chain, the TF is searching the DNA, undergoing 1D diffusion. It can stochastically bind to any site, specific or non-specific, but the specific sites are very sparse, okay? Compared to the non-specific sites. And the difference in the binding affinity is very strong. So the probability of unbinding from the specific site is much lower than the probability of binding from the non-specific site. So if you sort of create this cartoon model where you're thinking about just the kinetic effect of, so basically the idea is that the transcription factor can search around the DNA for variable lengths of time before finding the specific site and binding to it. And that's sort of what this is saying. And if you have that multiplicity of time scales, then you end up with a power law distribution of survival times. This is sort of one way of looking at it, but I think a more meaningful way of thinking about it is that there is a lot of heterogeneity, both in the nuclear environment, but also in the sequence strengths that a transcription factor can bind to, and this broad distribution might lead to a power law behavior. And then we looked a little into the physics literature, but I'll just give a little very quick snapshot over here. There was some calculation, some work by Boucha and George. I think they were looking at glassy dynamics. Maybe all of you know this better than me. But they were thinking about particles that are in sort of traps of different depths. And these trap depths can be distributed in some way, let's say exponentially. But then again, if there's a multiplicity of these trap depths, then the escape times can be modeled and actually calculated out to come out as a power law. So if you can think about the binding of transcription factors to different sequences and different areas of the DNA as being in different traps, one can again think that perhaps it makes sense to think about a power law distribution of survival. And so this is suggesting some sort of I have another set of just kind of talking about how can this then be related to gene expression. So they could be very different strengths of binding motifs, but also there could be conformational changes in transcription factors, specifically in their unstructured domains. And as Jay talked about condensates, there could be increased concentration within condensates. And all of these could change both the binding affinities and then the K-on and K-off. So with that said, how does one relate the transcription factor dynamics to gene expression? And I think that's an ongoing question with not necessarily very clear answers yet. But this was a very interesting recent paper, I believe from Gebhard Lab, where they made the synthetic transcription factors with multiple DNA binding modules. So some have 1, others have 4. But they also had the TF fused to this auxin degron system to induce degradation. So they could either change the number of binding domains or they could change the concentration. What they found was the effect of the number of binding, and then they looked at the residence time. So they found that the effect of changing the residence time led to much stronger change in the mRNA output compared to change in the concentration of the transcription factors. And so this is another example that suggests that binding time might be a critical determinant of transcriptional output. But this is an area of ongoing research that, is one of the things for the future. So Gordon's lab has also been working on looking at mRNA production, gene expression at specific loci. And this is this particular system that they created, this MMTV array, which basically is a GR-sensitive promoter sequence. And the idea is that whenever you see this bright spot show up over here, you're seeing mRNA production. And if you look at the intensity, and this is the bursting that Jay referred to. So it's called RNA bursting. Again, unpacking it here a little bit. So you see these intensities sort of on, off dynamics. over minutes to hours, so very long time scales. And then you can calculate the distribution of sort of on times and the distribution of off times. Yes. Yeah. That's a good question. Why are there? Possibly. Actually, that was the only frame where 3 showed. So they can have more, yeah. Yeah, these are these mouse carcinoma cell lines. They could have additional, I guess. Yeah, yeah. And so another knob that we have with the GR, ER, these hormone receptors, is that in the body, they often show a cyclic, sort of a time-dependent activation because they're these hormone cycles, everybody might know. So it turns out that early activation, which means if you do this experiment within 1 to 6 hours of ligand activation, led to longer on times and shorter off times, as you can see just from this distribution. The dotted line is the early activation, and the late activation led to shorter off times. So, whoops, sorry. And so the on times were of RNA bursting was ligand dependent. And then how could we try and connect it to TF dynamics that I showed? So then we can take the same system and now look at single molecule GR dynamics. And what we found is that the early activation led to longer dwell time. So basically, it had a shallower exponent. And the late activation, which was 6 to 12 hours, led to a shorter dwell time as shown by a longer exponent. So this is sort of at least consistent with the fact that early activation, which leads to long GR binding times, also leads to longer on times at the mRNA production level, whereas the late activation leads to shorter on times and shorter GR binding times. As you may notice, there's still a very big discrepancy in the time scale. The bursting is taking place over hours, minutes to hours, whereas the transcription factor dwell times are still on the order of tens of seconds. And I think this is a question that needs, there's several different models in the literature, which I don't have time to really go over, thinking about how these might be connected. That's super long. I feel that it shouldn't be super long. I think I know, maybe we can talk about that. Yeah, I think I know what you're talking about, but I have to remember which paper it was. Yeah, yeah. Yeah, so in fact, some of these models talk about, could it be that the transcription factor binding is just needed to initiate the whole process, and then it can come off, but then gene expression continues over much longer time scale? Or is it coming on and off? It has to happen more dynamically. Or, so exactly. So maybe these experiments might help to figure this out. Right. Yeah, yeah, yeah. And that absolutely is known. Yeah. And there's some recent tracking data. Right. This is just sort of the correlation that I already mentioned. There's some recent new modalities, new microscopes, such as the orbital tracking microscope, which might be able to look at transcription factor dynamic at or near a gene locus over longer times. That might help to answer these questions. So this is kind of a summary of this first part, where by looking at the dwell times distribution, it appears that there's a broad distribution of binding affinities, and there's still an outstanding question is what exactly is the origin of these? How might these be perturbed by changing the different properties of the transcription factors in terms of their binding modalities, as well as their ability to lead to productive gene expression. So then I'll switch to the second part in terms of how much does a TF move. So with these with these single particle tracks, we can extract that information as well. But just a quick overview that there has been a lot of interest in imaging chromatin dynamics at multiple scales. This shows the tracking of a single locus within a nucleus. This shows an example of tracking H2B. So going back to chromatin and how chromatin itself is moving inside the nucleus. This is single molecule imaging of chromatin, histones. And then a number of labs have looked at overall sort of the global flows of chromatin. And this was from Shaban et al. But the movie I'm showing you is actually a T-cell nucleus from our lab. And we've started working on this also, where you can actually overall see the dynamics of the chromatin. So to get a clearer handle on the mobility, we need to do two types of imaging, kind of fast imaging, as I mentioned, about 100 Hertz, to image all of the diffusive molecules and thereby diffusive molecules and bound molecules. And then from there, we can get the bound and the unbound fraction. But then the TFs are binding over tens of seconds to chromatin. So we wanted to look at the dynamics, the spatial mobilities of both chromatin and TFs over these timescales. So then we use sort of this slower SMT to do that. So I'll show you first what we found with histones. And so this is a movie of H2B, which is a core histone. It incorporates into the nucleus. And here we were using these exposure and intervals and taking about 2-minute long movies. This is similar to the movie I showed you earlier. You get the particles, you get tracks, and this is what the tracks look like. So you can see there's a plethora of track lengths and speeds and diffusion coefficients. This is sparse labeling, yeah. So then we turn to a technique developed by Simon Mokri at Yale, perturbation expectation maximization, to basically extract a set of diffusive states from a population of trajectories that has a wide multiplicity of diffusion coefficients. And this is the technique we'd use to look at the states of the B cell receptor molecules as well. Yeah. Yes, you're right. The laser's off most of the time. Yeah, yeah. So we will lose out on very fast molecules with this imaging. So therefore, we can't really quantify diffusion. So what we're looking at is the diffusive light motion. I don't want to call it subdiffusive because there's a lot of statistical physicists here, but sort of subdiffusive light motion, not necessarily free diffusion. So yes, with that, we're not going to be able to resolve the fast diffusion. Yeah.
Did you try to just
Oh, like I said, we do two types of imaging. I'm just showing you this one. I guess in hindsight, I should have put a movie. So we also do fast imaging at 100 Hertz. So 10 millisecond continuous exposure. No, what I was showing you was the slow SMT, but we do fast SMT also on the same type of samples. And then we obtain the bound and unbound fraction from there. And then I'll tell you what we do with that. But then we specifically or purposefully, we slow down the imaging so we're able to look longer at the ones that are not bound, because otherwise they'll photo bleach too fast and we'll get very short time. Does that make sense?
I think so. I mean, I guess you're fast, 10 milliseconds.
Yeah.
But it's space 200 milliseconds.
No. It's 10-10. A 10 millisecond exposure continuously. Sorry, yeah, I'm realizing some. These are halo tag. Yes, yeah.
Goes in the inter, it stays. There's supposed to be
Shay is not here. He knew about halo tag better. I think it's long lived. I think it's very long-lived. I just don't know how long, but as far as I know, forever. Okay.
The kids are
Yeah, so they photo bleach much faster than they'll ever. But also, as we can see, there's this light sheet that we have. So we're looking at molecules that might keep coming into that plane, and we look at one cell for a few minutes, like, you know, for one movie, then we go to another cell. So what we have to do is actually assemble tens of thousands of trajectories. And so we collect data from many cells to actually get that information. So then what we do with this technique is to look at the so we are getting longer tracks, but then we look at subtracks that are 7 frames long. And what this algorithm does is to calculate the posterior probability of a subtract belonging to a certain diffusive state. And so from here, so you can see, you know, this is the lowest mobility and highest mobility. So we're getting the information about the diffusive state at the subtract level from longer tracks. So this is in part to be able to get the local information, but also there could be transitions within these long tracks. They could be transitions on the type of mobility. And so we wanted to sort of, this is one type of analysis that we're doing. So what happens when we do this, that we can take the tracks, classify them, and the program itself is agnostic to the number of states. So you can ask it to fit it to as many possible states. But what we find, for example, with histones, so the histones are also moving. And that was the point I wanted to make in that previous slide, that while they're part of the chromatin itself, they're moving. But what is their movement like? And we see that they fall within these sort of 2 low mobility states, which we'll call 1 and 2. One, a very low mobility, and then a slightly higher mobility state. And interestingly, there's some amount of unbound fraction. So these experiments, there's some unbound histones in the nucleus, and typically people find within somewhere between 10 and 30% are unbound. So we focus on sort of these 2 low mobility states. What we found, interestingly, that if we look at the overall track level now and stitch back these subtracks into the longer tracks, within a longer track, the molecule appears to switch between a low and a high mobility state. So it's like there is a long track, but it's going back and forth. The same molecule is going back and forth between being very slow and not so slow. And so this was kind of interesting and surprising. And this shows the 50 longest trajectories we have. And this is like laying it out and the line stretching out your trajectory and color coding it, with the red being the lowest mobility state and the blue being the slightly higher mobility state. So you can see that there are these long stretches of red, but there are occasions where it switches into sort of this blue state. And this just shows that there's a greater probability of just staying within a state when there is a particle that is in one state. But they prefer to remain in the same state. Probability of, the diffusion coefficient. So that kind of is here. So it's moving about 2 microns in 1 second, approximately. No, we can't capture, like diffusion, and I don't have that, but diffusion is much faster than this timescale. So if it's freely diffusing, then it's just going to disappear most of the time with this 200 millisecond imaging interval that we take. Yeah, so we pretty certain that these are bound all the time, but they're just showing 2 different types of motion.
Yeah, spatial. That's states that tends to be more stately from spatially distributed.
We haven't seen that yet. So we did try it with the data that we have. But and that was sort of one of the first questions. Are they more strongly localized to heterochromatin versus euchromatin, for example? And that is our ongoing study to do. That's why I was asking you the 2-color type of imaging. So we can look at histones, the heterochromatin with a different color. But with the data that we have, we did not see a spatial. But I'm not sure that that's absolutely that we have, because we need somehow have to figure out a way to get, because we are getting only a few number of tracks per cell. Then how do we superimpose many, many different cells to really know that we're right? Yeah, and that's on our mind. So in Gordon's lab, they actually did use the PA JF dye. And yeah, we've used it for some experiments, but we need to do many more. So yeah, I'd be happy to talk, because I know you showed this clustering of enhancers, right? Yeah. So it would be really good if we could look at some sort of spatial map like that. Yeah, yeah. And then, so this was what I said. So what happens to the transcription factors if we look at the same type of analysis with GR, for example? You know, how do they do, you know, what kind of mobility states do they show? So this is now showing GR, which looks exactly the same in a way, but you actually see a lot more of the molecules sort of disappearing and moving around, more of the higher mobility states. So again, we see 2 low mobility states, 1 and 2. And very interestingly, they're more or less the same as the histone states. So the dotted lines are histones and the solid lines are GR. And here actually you see this green one is something somewhat more mobile. We don't know, but it's a very small population fraction. Yeah, yeah, I think it's not really curvature. I tell my students they shouldn't join the dot on the 0, but they do. And I mean, I like, should be doing, like, it's hard to say from, so it's hard to, if you leave out a 0. And that's why I said I hesitate to call it subdiffusive, because we just don't, with the subtrack level, we just don't have enough timescale information to know whether it's subdiffusive or not. But I'll show you MSDs for the longer tracks. But here we're just calling them. Yeah, yeah. We are using Nikon, but Gordon's lab, they use Olympus.
Yeah, so what happens, what we find, we also see this, the first time point is weird, and then we go back and we look at the time. The time is longer for the first year.
Really? We haven't seen that.
Yeah,
But
you
no.
should, dig into the files. Probably, or we figured out this whole thing. We also see this weird shape.
Right.
Just the first thing was taken.
But that doesn't matter, because for MSD, right, we're looking at all delta T's that are 0.1 second or 0.2 second, right? All across the track. So the first frame is just a very small part of it. So I don't think that matters. Yeah, yeah, yeah. So anyway, so they look very similar to histones. And then also they show switching between states, you know, the tracks themselves, as you can see here in the example track. And also these are some of the longest tracks that we stitched together. So you see a lot of state 1s, and then, but then there's some jumping back and forth between state 2. What does this mean? I'll get to that. But again, multiple receptors, steroid receptors, ER, AR, PR, they also showed very similar state 1 and state 2 in terms of the actual mobility or diffusion coefficient as H2B, and also the switching between. But there's some differences in, for example, AR and PR, you see many more of the state twos, and we have some thoughts on why that might be the case. And this is showing other co-regulators, GRIP1, MET26, Relay, CTCF, etc. So the picture is emerging that the chromatin is moving in these sort of two states. Anything that binds to it binds to chromatin, and so it moves, and it shows the same thing. Yes. You mean this one? Yeah, I feel this is not something to, because literally there's not anything at 0, right? It's basically starting at that first time like is 0.2. So there's nothing there. I don't know, the reviewers didn't ask us to remove that point, but I feel like, is that wrong to put that point there? I don't know. Right. Yeah, so it kind of should be there, but yeah, I don't know. Does this have a meaning? I don't know. Maybe we can discuss it. So the physical picture is that there's these two mobility states of chromatin, but also of transcription factors. So then we wanted to look a little bit more detail into the biological origin of these states, and then the different domains of the transcription factors and if they're needed. And so the first test was just looking at GR untreated. You still have some GR molecules that are in the nucleus that one is able to track. And you kind of see the two states, but the state 1 is almost non-existent with untreated compared to the GR that is activated with DEX. So hormone activation increases the proportion of this lowest mobility state. And also we find the same thing for ER, PR, and AR. You see a much greater increase in state 1. This is a fold change upon hormone activation for state 1, that lowest mobility state. And then we looked at mutations. So basically mutating the DNA binding domain, the C428G at basically one of the sites. It's shown with ChIP-seq, for example, that this mutation, there is no binding to DNA. You don't see any ChIP-seq peaks. And also when we do the SMT analysis and the mobility, we see that there is almost no low mobility 1 state. And a similar outcome is observed by washing out the hormone and another mutant which also doesn't bind chromatin. So this suggests that state 1 is related to specific binding of the transcription factor to DNA. And another test was that MMTV array that I mentioned earlier, which basically it serves as a probe for chromatin binding. And when we look at GR within the array, you can see the array here, as opposed to outside the array in the nucleoplasm, we see a lot more of state 1 within the array for GR. So basically that array allows multiple copies of GR to bind. So again, this chromatin specific binding state appears to be enriched for state 1. We then turn to another transcription factor, the peroxisome proliferate activated receptor, gamma-2. It binds chromatin as a heterodimer with this RXR heterodimerization domain. It's also modular. It has an activation function domain, a DNA binding domain, so these can be mutated. And wild-type PPAR gamma shows again 2 distinct low mobility states, state 1 and state 2, which also shows switching, again, a lot more in state 1. So we did a positive sort of test where we overexpress CEBP alpha, which facilitates PPAR binding to DNA, plus these other mutations. And this just shows that there is indeed CPPP assisted PPAR gamma loading as found in ChIP-seq. So it does help in binding. And so when we did SMT in a cell line that had overexpression of CBP alpha, we see increased state 1, okay, and a decrease in the low mobility state 2. Then when we looked at the DBD mutant, the DNA binding mutant by itself, Then we see a lower and a decrease in the state one, and not that much of a change in terms of state two. Then we look just at the heterodimerization mutant, and we see again a lowered state one. And the double mutant, where you mutate the DNA binding domain and the heterodimerization domain, and basically that appears to get rid of all the binding in this lowest mobility state. So using p-power gamma 2 also, it appears that this lowest mobility state is related to specific binding. So what about the second low mobility state, sort of this, the second slightly higher low mobility state? What might be the role of TF-TF interactions, IDR mediated? So we just have some clues on this, but we're working on this more. So GR contains a long intrinsic disordered region and has also been shown to lead to formation of condensates. This is an example showing condensate formation. We haven't explored the relationship between condensates and the single molecule imaging, so this just shows that they can form condensates. But then complete deletion of this domain actually led to loss of state 2. So most of the SMT results showed that they were in this chromatin bound of state 1 that we had observed. But this was a very strong sort of blunt hammer type approach, removing the whole thing. So there's work ongoing in Gordon's lab where they're mutating more specific mutations in this IDR region to have a more fine-tuned, to get a better, more fine-tuned information about how this affects the mobility. And so this is just a reminder that we found that GR dwell times are power law distributed. Interestingly, when we look at the dwell times for this IDR mutant GR407C, it appears to follow better an exponential, a bi-exponential distribution. So it appears that the loss of IDR leads to the loss of power law. So what then could these, in part, IDR interactions, or maybe other interactions that we haven't quite probed, be in part what is leading to these power law distributed binding times? And so motif analysis also showed that the log odds distribution of these motifs showed that in this N-terminus, the IDR mutant, which shown in pink over here, has a narrower motif distribution than GR wild type. Kind of inconsistent with our hypothesis that the broad distribution, the power law distribution, in part emerges due to broad distribution of binding affinities. So then we switch to another way of analyzing the tracks. I think I have 5 more minutes, maybe. So we look now at the overall tracks, not at those small subtracks that I mentioned. And we looked at the Van Hoff correlation, which is the step size or jump distribution. And then we can use a number of sort of Gaussian basis functions to then extract the distribution of MSDs that will lead to this jump distribution. So it's an iterative algorithm which was developed by Richardson Lucy, actually for denoising of images in image analysis. So when we do that, we see that the MSD distribution that leads to the observed, empirically observed jump distributions shows 2 lobes, right? So it's showing that even at the track level, there is 2 types of overall track mobility, a lower mobility and a higher mobility. And we're going to call these groups and not states. So RL group 1 and RL group 2. And we then went back to these tracks. So these are now whole tracks that move less, or and whole tracks that move more. Then on those tracks, we looked at what the PEM states, the little subtrack level information is telling us. And so we made these sort of heat maps of the switching kind of red-blue states. And we see that the ones that are moving less, on the left, have mostly state 1. So they are basically staying in this state 1, whereas the tracks that are moving more overall are composed mostly of state 2, with quite a bit of switching also back and forth with state 2 and state 1. So again, this is the picture that while we got some information from the subtrack level, it's telling us that these lower mobility tracks are the ones that are in state 1. And then going to MSDs, interestingly, we see both of these states, state 1 and 2, sorry, the resolution here is very low, show a slope of, are subdiffusive and show a slope of 0.3. There is a small fraction with a higher slope of 0.5, and these are sort of the slightly more diffusing particles. And there's been a lot of models in the literature, polymer models of chromatin in the nucleus. And typically a Rouse model of a polymer chain shows square root dependence on time. So one would expect 0.5, but we're consistently getting 0.3 for these two states. So this is suggestive of longer range interactions in the chromatin that might lead to this. And this is just an example showing, some studies in the literature where they look at the dynamics of chromatin in the nucleus, modeling it as a self-avoiding heteropolymer. And then they can introduce these CCF cohesion loops as extra terms in the potential energy. And what they find is that it does lead to a lowering of the MSD exponent, kind of to 0.4. So but we're finding 0.3 and also in some other data sets. So I think that's kind of real. So that's something interesting to think about, what's leading to these highly subdiffusive motion that we're observing. And very quickly, there's some additional experiments that were done in Gordon's lab to look at the role of cohesion and how cohesion and GR interactions are mediated. But just very quickly, I wanted to point out that knockdown of cohesion, also using this Degron system, leads to increased H2B mobility. So much lower state 1, but higher state 2. And then if you look at GR in those systems, in the cells in which cohesion has been knocked out, you actually see that it decreases the binding. So you get less of the bound fraction of GR. So this is, again, kind of conflating things. This is space, this is time in a way, the binding fraction. But we are looking into further how cohesion knockdown itself, as well as mutations in one of the cohesion loader proteins, NIPBL, how this changes the states, the mobility states and binding times of GR itself. So we are, there's ongoing work on that. But this is suggestive that is it the increased nucleosome mobility that might be leading to the decreased transcription factor binding, the GR binding that we observed? It is, yes. And this was a paper that they showed that, yes, that GR binding is very intimately related to cohesion. Yeah. And they're still working on figuring out more fine-tuned details. And finally, you might be thinking, how can we then relate these mobilities with binding and timescales? Because we have the space and time. So this is a preliminary attempt at that. So we can take those two RL groups that I showed you, those overall tracks that are slower moving, faster moving. And then we look at their survival probability. So this one is showing the lower mobility group. So the black shows H2B distribution, and the purple shows either GR or ER distribution. So the lower mobility groups kind of look similar to histone distribution. They're sort of long-lived, basically, indicative, again, of strong binding. But then if we look at that higher mobility group, the other group that we sorted out with the RL analysis, because we can actually do survival distribution from there, we see that they seem to follow a power law pretty well. So this is starting to give us some hints that while both of these states are bound to chromatin and appear to follow chromatin dynamics, there's differences between them in terms of how they associate with chromatin. You have more or less association, but also their binding times may be strongly correlated with how much they're moving in space. And this is a recent paper from Tim Stasevich's lab, where they used an antibody to label regions of the chromatin, and this is getting to your question a little bit. So these purple lines show H2B within enhanced H3K27 acetylation, which are like poised chromatin. And the green shows RNA polymerase 2 with serine 5 phosphorylation, which are actively transcribing. Then they looked at histones inside these domains and outside them, and basically found that the H2B mobility was enhanced within these regions compared to outside, right? But here with the actively transcribing, these H2B showed lower mobility. So then it's again suggesting that active transcription is leading to or associated with lower chromatin mobility. And the question then is, what are these regions doing to transcription factors, if you're able to look at it? So hopefully with new imaging techniques that we're trying to establish, we'll be able to do that. And finally, there's a model from Dave Thirumalai's group where they actively put forces in different parts of the chromatin, heteroactive locus versus inactive locus. So they had force dipoles in regions of the chromatin. And what they found with the simulations was you see overall lower MSDs for loci that have forces on them, that are actively transcribing loci. And these match experiments that were done by Mishima's group, in which they inhibited transcription with some small molecule inhibitors, and they also showed that overall chromatin mobility decreased when you had active transcription. So with that, I just wanted to give my final summary that with this type of analysis, we saw both histones, transcription factors, and co-regulators exhibiting two distinct low mobility states. And while the low mobility states of TFs match those of H2B, the fraction of time that a TF binds, or the population fraction of TFs binding to one chromatin state versus another, the propensity to bind seems to change and depend on its activation status, where state 1 correspond to specific DNA binding, state 2, we don't know yet, non-specific interactions, IDR interactions, TF interactions. And we have been trying to think of a physical picture of this, but one of the analogies that comes up is a boat in an ocean. The ocean itself is moving, then you have a boat in there, and then you have people in the boat. So you can have like little local movements, but then all of that is going to be constrained by the overall sort of movement of chromatin in the nucleus. And finally, some open questions are, we are still working on understanding the origin of these states. How do these relate to and correlate with transcription, trying to do co-localized imaging of transcription factor dynamics along with mRNA bursting, for example. As you asked, how are these spatially organized in the nucleus, how these relate to binding times. And finally, I hope we are close to answering the question that we had asked a few years ago as to how physical cues regulate the dynamics of these transcription factors and gene expression. With that, I want to thank you and also thank the people who've done this work. These are the current graduate students in my group, a few undergraduate students. The work on transcription factor dynamics I talked to you about was done by David and Kostab together with Gordon Hager's lab and many collaborators on different projects, funding sources. And if people are interested in postdoc or graduate student positions, please contact me. Thanks very much for the questions and attention. Happy to take other questions. Yeah. Right. You're actually right. We're going in the opposite direction in those because they're from what was considered two states. We're saying there's this multiplicity and this broad distribution. Here we're kind of seeing a broad distribution, then we're saying they're two states. Yes, absolutely, it's possible. And I think it's sort of a constant struggle. Even the method we used is a probabilistic method. So we put something in state one, but it just says that the probability of it belonging to state one is 60% or more. And we put something in state two, the probability of it belonging. So within it, yes, it's not sharp peaks. There is a distribution, but what we think is the distinction is strong enough to identify as two states. Yeah.
Specifically wondering the case where
Yeah.
you said the second stage is called. How do we interpret that?
Yeah, so with a grain of salt. And that's why we're doing further experiments with better mutations of the IDR. And in fact, what I didn't say is we didn't represent anything that was less than, I think, 5 or 7%. So it's possible that there is some of that state available there, but also possible that some of the ones that were in state two got misclassified. So that is possible. So I would say that having a black and white picture is not possible, but I think the two shades of gray are distinct enough. Yeah, thanks.
I mean, how do you think that, like this PEM model that you use, like all relies on the fact that you have purely diffusive motion that like P is linear, like everything is linear with time?
So it actually accounts for non-anomalous diffusion as well.
But then how do you compare diffusion constants from like tracking different numbers?
So yeah, the key there is we're using small tracks. So I think it's not possible to really distinguish anomalous diffusion versus pure diffusion with such small tracks. So over those time scales, I think it's an okay assumption to assume that they're diffusive. And just based on the analysis, also you have this covariance matrix, and then you would get, I believe, more non-diagonal terms if you have anomalous diffusion. But again, with these small tracks, we won't be able to distinguish between anomalous and yes, I agree, yeah. And so we're not. Which is why I said I don't know. Are they subdiffusive or not? They're just very slowly diffused. Yeah. So I think one needs longer time scale data, which is what then we did in the other analysis where we didn't use PDM, where we had the whole track length. From there, we see that at the track level, they do appear subdiffusive. Thank you.
Yes. Yeah. I think it's about time.